Thank you, Ethan. All right. Hi, everybody. I'm also a co-chair of the public lecture series. My name is Vanessa, and I'm also a third year in the Neuroscience graduate program. Our first speaker today is Dr. Doug Coulter. Dr. Coulter is a professor of pediatrics at the Perelman School of Medicine at the University of Pennsylvania and a neurology researcher at the Children's Hospital of Philadelphia, CHOP. He originally received his PhD from Boston University's Marine program in Woods Hole, Massachusetts. Because a PhD isn't enough, Dr. Coulter moved to California, where he continued his postdoctoral training in Dr. David Prince's laboratory at Stanford University. Throughout his career, his research interests have focused primarily on trying to uncover the cellular and molecular mechanisms that govern epilepsy, including what exactly are anti-epileptic drugs doing in the body and brain, as well as investigating different types of epilepsy treatments in order to truly be able to increase the development of enhanced therapeutic strategies. As a neuroscience graduate student, it has been an absolute pleasure to have Doug as one of my professors in one of my first courses here at Penn, and I'm so excited that everyone today also gets to experience this amazing scientist and his work. Please help me welcome Dr. Doug Coulter and the title of his talk, Hippocampal Circuit Dysfunction in Neurologic Disease. Take it away, Doug.
Thanks. Let me see if I can get my screen shared here. Oh, okay. So it's this. Let me see what we get. Okay, there we are. Sorry, everybody. This crowdcast platform is a little difficult to manage. Can everybody see my screen?
Everything looks perfect.
Fantastic. So let me start my timer. Thanks very much, Vanessa and Ethan, for the introduction and the kind words, Vanessa. So I am going to be talking to you, as Vanessa said, about hippocampal circuit dysfunction and neurologic disease. And really, in the context of the hippocampus, we're going to hear all about some specializations and properties of the hippocampus that endow it with its ability to store and remember. And those same properties that endow it with this tremendous property that helps define us as humans also make it exquisitely fragile. So they tend to make neurons more prone to excited toxic cell death. They tend to promote aberrant plasticity. And what we end up seeing is that the hippocampus becomes a prime culprit in many dysfunctions we think of clinically, which include psychiatric and neurologic disease. And today I'm going to be talking to you about epilepsy in particular. And according to the Institute of Medicine report, 1 in 26 people will develop epilepsy over their lifespan, which is a pretty astounding statistic.
Hi, Doug. Sorry to cut you off. We can see your slides, but it's not in presenting mode.
Okay.
So we see everything, just not the whole slide as what we'd like.
All right. So on my screen, it's doing that. So still the same? Is that still the same?
Sadly. It looks like maybe you're presenting like a different window, or like the window that pops up when you're trying to fully present, like presenter mode.
All right, let me try this again. Where's YZ? YZ is the fallback genius on all this.
YZ, would you like to jump on? Oh, that's perfect.
It's okay now?
Yeah. Perfect.
Okay, it looks exactly the same to me, so it's unclear what's going on. Anyway, I apologize for that. So it's a prevalent disorder, and it's defined by a shared symptom as seizures. But but defining a set of diseases by a symptom is problematic. It's really a constellation of disorders. So it's like saying I have a sneezing disorder, right? You can sneeze for a lot of reasons. You can have seizures for a lot of reasons. And seizures themselves are defined as excessive synchronous discharge in populations of neurons. So epilepsy is primarily a function of aberrant neural circuits. Some forms of epilepsy are acquired, meaning that some kind of insult or experience that you've had generates the epilepsy, and some are genetic. And the most prevalent form in the adult population is mesial temporal lobe epilepsy, which Ethan introduced. In addition to the seizures characteristic of the condition, there are significant cognitive and affective disruptions associated with the TLE. And patients really dislike these comorbidities and express that they may be among the most disruptive symptoms of TLE, even more than the seizures themselves. And this is a disruption in circuits, as you might imagine, the same circuits that are supporting cognition and affective behaviors are also disrupted. And so they may be generating seizures, but on a tonic level, they're also disrupting normal functions of the hippocampus. So as Ethan alluded to, the therapy for TLE is surgical resection of the focal hippocampus. This happens quite frequently because many patients do not achieve adequate control by conventional anti-epileptic drug therapy. And so this is a very successful treatment in terms of reducing seizure burden. However, significant subsets of these patients after the resection exacerbate their verbal memory deficits. So they already have deficits and they get a little worse after the resection. And this is true for both pediatric and adult cases. The pediatric cases frequently get better because there's a lot of plasticity in the developing brain. But really the critical questions for me and what's driving this talk and this line of research that's been going on for several years in my lab is the hippocampus so damaged that normal behaviors supported by this region are irrevocably lost? So that's what you might expect and hope for, actually, if you're going to take it out. Hopefully the function is gone and you're not taking out functional tissue. But a possibility remains in that there may be residual masked function in the damaged hippocampus, which can be targeted for therapy to improve seizures, cognition, and emotional comorbidities. And so that's kind of one of the things we're assessing in this talk, and it's going to be the main focus of the talk. Again, surgical resection is unilateral. We learned that from HM. HM actually had an epilepsy surgery, and he had a bilateral hippocampectomy with this devastating consequences. But up to 80% of patients who receive a temporal lobectomy when they've been adequately worked up get seizure-free or have their seizures frequencies reduced significantly. So it's an effective therapy. So we've heard a general overview of the hippocampus, but I thought I'd introduce you briefly to the synaptic anatomy of the hippocampus. The entorhinal cortex, here termed EC, provides input to the hippocampus through the dentate gyrus. And this is the perforant path. The dentate granule cells, the principal cells of the dentate gyrus, receive this input, get excited, and send their axons onto downstream afferent targets, area CA3, and then CA3 projects onto CA1, and then CA1 closes a loop by projecting back to lateral entorhinal cortex and also to the subiculum, which projects to the entorhinal cortex. So this is a series of excitatory connections that form a loop. And in engineering terms, that's an unstable situation, and that may be one of the reasons why this structure is so prone to seizures. I'm going to focus our attention on the dentate gyrus in this talk, and I'll tell you why in a minute, and then get to the data. The dentate gyrus has a critical role in contextual encoding and also in anxiety, and both of these are disrupted in temporal lobe epilepsy. Among the various hippocampal subfields, the dentate exhibits the most significant disruption in excitability. And I'll show you this in temporal lobe epilepsy models, and also in tissue resected from patients with epilepsy. And there's also very large scale changes in the anatomy and connectivity in both TLE models and in tissue resected from patients with epilepsy. And these combined factors make it an ideal therapy for circuit targeting to try to control both cognitive and seizure disruptions. So a main property of the dentate that kind of defines its function is that it activates very, very, very sparsely. And this is due to a lot of factors, but one of the main ones is local circuit inhibition within the dentate gyrus. The anterolateral cortex, as I discussed previously, projects to the dentate through the perforant path, the medial and lateral perforant path, excites these granule cells, and those granule cells then project on out to CA3. However, in this color-coded diagram depicting its activation, you can see that this is the false color for how many action potentials are being fired per unit time. You can see that the anterolateral cortex is a very excitable structure with a lot of warm colors in this sparse population of neurons that's activated. These projected granule cells, most of which do not activate, they do not fire in response to this massive input. Very rarely does a granule cell fire, and then at very low rates. If you look at the false color scale, and then it projects its output onto CA3. This sparse network is central to the function of the dentate gyrus, in that one of the main things that it does is called sparse coding, in that it can discriminate different inputs based on which cells fire. And this is really important, and an important cognitive function called pattern separation. And pattern separation, you can think about, say, if you go to the mall. I know we haven't gone to the mall in a couple of years, but if you go to the mall and you go to the mall regularly, and you're a creature of habit like me, you park your car in the same area of the parking garage every time, but then you can go and find your car again on a daily or weekly basis, separated by time, despite the fact that you're in a very stereotyped place with all the same cues, with very, very little difference. And that your ability to do that depends on you having a functioning dentate, that you can separate very similar patterns into distinct outcomes. So the first thing we wanted to do is look at one of our animal models of epilepsy and begin to look at whether this critical property of the dentate was maintained or corrupted in epilepsy. And we chose to do this using a circuit imaging technique called calcium imaging, multicellular calcium imaging. And these calcium indicators are loaded into the tissue, into the cells themselves. And when cells fire an action potential, they blink. There's a flash. And it's a way to code large populations of cells and determine their firing properties. And so we chose an animal model in which we inject a convulsant into a rodent. This convulsant induces a severe seizure, which in turn injures the brain. The animals recover, they're nursed back to health, and then weeks to months later, they begin to develop spontaneous seizures. And those seizures originate in the limbic system, and there's an induced pattern of hippocampal injury that's very similar to that seen in TLE patients. If we go and assess the function of these granule cells in this model, or before injury and at various time points after injury, what you can see is that in a control situation, when we stimulate the afferent, the perforant path to the dentate granule cells, very, very few cells activate. However, immediately following an injury, and then permanently, what we see is that there's a loss of this selective activation, and the entire network will activate in response to simple afferent stimulation. And so we've lost the sparse activation properties dramatically. And this is plotted here at varying time points after the percent of responsive granule cells at these varying time points. Importantly, if we block inhibition, the local circuit network of inhibitory neurons, all these time points activate the same. So all of the cells are able to be activated, and inhibition defines who can activate. This suggests that disinhibition is occurring in the epileptic dentate gyrus. We have many lines of investigation that support this, but I don't have the time to talk about them today. In addition to these functional changes with this loss of sparse activation, there are anatomic changes. The axons of granule cells also contain zinc, and so you can use a photoemulsion technique to highlight where the axons go. These are the granule cells here. This is where their axons go, this dark color, and you can see they target the hilus and CA3. However, in the epileptic model, and also in human tissue resected from patients, there's this extra layer of dark emulsion staining, which reflects the fact that axons of granule cells sprout and re-innervate aberrant targets. So there's a local network that wasn't there before in the epileptic brain. And in these Nissl stains below from the same tissue, you can see that many of the neurons present in the hilus and in CA3 are die out or are thinned out in the epileptic brain. So there's significant neuronal loss. And this is plotted below in epileptic pilocarpine mice and in controls. So to summarize all these changes, in this animal model and in humans with epilepsy, there's an initial injury. And in rodents, weeks to months later, and humans sometimes up to a decade later, seizures emerge. And early after the injury, there's this cell death, which I've highlighted for you. Later, the GABA receptor repression begins, the disinhibition. And there's gliosis, significant gliosis, which also disrupts circuit activity. And then there's aberrant neurogenesis, as Amelia Aish is going to be talking about roles of neurogenesis in psychiatric disorders, but it plays a role in epilepsy as well. And then you have these circuit rearrangements, the mossy fiber sprouting at these varying time points after injury. And then the inhibition is reduced chronically across the whole time frame. And the activation of granule cells, normally sparse, is maintained throughout. So this is the constellation of circuit changes that we can see in the epileptic dentate gyrus. Now, the questions I'd like to address, given this background, is that first, is there evidence that epilepsy-associated dentate gyrus disruptions generate cognitive comorbidities? And if so, are these comorbidities reversible? So should we be taking the dentate out if it has maintained function? And if we leave it in, is there something we can do to block this aberrant function? And so Julia Kahn, who is the student who did the majority of this work and is also one of the first GLIA students who helped start this whole speaker platform, came up with a way to assess pattern separation, which is called the spatial object task, spatial object recognition task. And in this task, you have multiple objects. So you're getting into a pattern separation context. And animals become very familiar with this task by being exposed to this, this box with the objects several times in a training motif. And then 24 hours later, they're asked to recall which of the objects moved, to figure out which of the objects moved. So they have to recall where it used to be. And then you look at how the mouse attends to the moved object relative to the stationary objects. And the second part of this is going to be, how do we reverse these epilepsy-associated dentate granule cell excitability? And the answer is using DREADS. And DREADS is proof that we sit in dark rooms and come up with weird acronyms all the time. And the DREAD acronym is designer receptors exclusively activated by designer drugs. And these are engineered receptors, in particular muscarinic and kappa opioid receptors, in which the binding pocket for the neurotransmitter is mutated, so it'll only respond to a different drug than the initial acetylcholine for these, or opioid receptor for kappa opioid receptors. And so these designer drugs are clozapine N oxide, or CNO, or salvinorin B, or SB, as I'll show in the further slides. And these activate the receptors. They have what would be a normal function, except only in the presence of these designer drugs. And HM3-DQ, the human muscarinic receptor, triggers depolarization. HM4-DI triggers hyperpolarization in these neurons. And the cords also trigger hyperpolarization. And so these can be expressed virally, so you can put the viruses in stereotaxically and target excitatory neurons and be able to specifically modulate granule cells. And just as in the interest of time, I won't focus on this, but here in a control mouse, here are the three objects. One of them gets displaced in the test, and this is the time the animals spend attending to the object. And you'll see that the displaced object got much more attention than it did before it was displaced. And that's shown in the false color here. In contrast, in the epileptic mouse, here's the background, and then this is the displaced object. The mouse does not pay any more attention to the displaced object than to the other objects. So the pilocarpine mice seem to have a specific deficit in this pattern separation task. And so that's shown here in a graph of the data. And here in a wild type mouse, the animal discrimination index indicates that it spends much more of its time investigating the moved object than it does the remaining objects. In contrast, the pilocarpine mouse, the epileptic mouse, doesn't attend to the moved object at all. And so it shows a very specific deficit in this pattern separation task. What happens when we reduce the hyperexcitability in this circuit using these designer receptors? And the answer is you can make the mouse smarter. So here's that pilocarpine mouse in which the vector contained GFP, i.e. no dread, and CNO is exposed, and it similarly cannot attend to the moved object. But when we reduce the hyperexcitability in the dentate granule cells using activated HM4DI, the animals attend to the displaced object as much as the controls did. So we can make these epileptic mice smarter, and I'd like to emphasize that this is only the dentate gyrus in the dorsal hippocampus that's getting modulated. Nowhere else in the brain is there any of the dreads expressed. And so this is a very, very specific intervention in granule cells. What about control cells, control granule cells? Well, control granule cells in control mice, they're able to discriminate very well. So here in the two GFP CNO traces, they can discriminate the moved object from the stationary object. But if we mimic epilepsy and increase excitability of the granule cells using activation of HM3DQ, the depolarizing one, you can see that these animals no longer can make that discrimination. And similarly, if we treat them identically to how we did the epileptic mice and decrease activation using an inhibitory dread, HM4DI, these animals also can't make the discrimination. So there's a window that enables these granule cells to function normally. And in control mice, it's in the realm of what they usually do. And in epileptic mice, it's in a context where they have to be, excitability has to be reduced. So we can restore the function of these networks. Finally, it could be a trivial association where maybe we're just blocking seizures and the animals are able to just function better if their seizures are being blocked. And that's not the case, because here are seizure plots for 3 mice. Here's the seizures per day, and this is over 3 months. You can see that epileptic mice tend to have seizures in clusters separated by about a week. And these seizures themselves were not affected by activation of an inhibitory dread. So what do we think is going on? Well, under control conditions, this sparse population that activates is very important in terms of having high information content, and is also very important in behavioral performance. And so these excitable cells generate the behavior, where the background granule cells are low in excitability, they don't activate, and they have little or no information content and don't affect behavioral performance. In contrast, in the epileptic brain, you still have this coding network, which we can get to reemerge in dread contexts, but there's this background hyperexcitability in the non-coding network. And so this background hyperexcitability is disrupting behavioral performance. And when we take the dreads and we push these networks back to more normal behavior, we can make the coding network reemerge, slide to the left in terms of excitability, and we can make the non-coding network slide to the left as well and become quiescent, and function can emerge. In contrast, if we activate the background network with HM3DQ, we can lose function and make the network non-functional. So to conclude, there's an appropriate window of excitability in which these circuits can code information, and that's critical, and that this is disrupted in disease states. But the disease-disrupted circuits still retain their underlying ability to code information. We can make them work by using this dread strategy, and there are other strategies I can envision that would also work. Despite this is despite the significant structural damage I showed you, the loss of these populations of cells, the sprouting of these axons, and also this loss of inhibition that's present. And so this might be a viable strategy for both seizures and also for generating a retention of cognitive abilities in these mice. The intervention is neuron-specific and modular, and this suggests that strategies to preserve hippocampal tissue but reverse excitability deficits might be a viable alternative to hippocampectomy. Last and most important, I'd like to highlight who did the work and who paid for the work. Julia Kahn was instrumental in this work, and so as I said, she's fantastic. She's moved on to industry, but she's just a wonderful person and a great scientist, and I was privileged to have her in my lab. As I said, she's one of the early members of the GLIA network that helped start this. Chris Stengler is also an NGG grad student who did quite a bit of the work on the calcium imaging. Russ Port was an NGG grad student. He was a postdoc when he was with me. He did some voltage-sensitive dye imaging for the studies. Haji Takano was just a valued colleague and partner in crime in my lab. And Chris Adam is the NGG grad student who's kind of taken the mantle and is continuing on this project. And Alicia White is a technician who generated the animal models. Funding was from the NIH, multiple sources, and the American Epilepsy Society. And thanks to Dave Meany and Brian Roth for their help with what we were doing. Thank you very much for your attention. I apologize for the delay early on in getting this session going. This I shouldn't be showing. There we go.
Yay, good job, Doug.
Thank you.
Okay, so it looks like we got like 3 minutes for questions, Q&A. One person, Dolly, asked, I guess on slide 8, is there a reason why day 14 looks like the control, or is this just specific to that recording?
No, that was a real finding, and it was kind of a push-pull. I got we don't know why it's happening. It occurs in both inhibition and also in the granule cell activation. And it seems like there's this failed effort by the network to compensate for the hyperexcitability. And it tries as best it can, and then it collapses. But that's me anthropomorphizing just a scientific finding. But it's a real thing, and we've seen it consistently.
Okay, YZ also asked a really good question. For human patients, do seizures typically start in the dentate gyrus? If so, does removing the dentate always impact the ability of a patient's ability to pattern separate?
So I don't think it's possible. It's possible to lesion the dentate. That's been done experimentally. I think there's a number of strategies that can do that, but I don't think it's possible to take it out without damaging everything else so much that you think that you've lost it. But when you lesion the dentate, pattern separation abilities are degraded. It's not entirely lost. Other circuits can contribute to pattern separation. But it's been shown in patients with epilepsy, with temporal lobe epilepsy, that their pattern separation capabilities specifically are severely disrupted. And I don't think I specifically answered your question, YZ, but I generally answered your question.
Thank you. And I'm sorry, Ethan asked a really good one. What are some strategies we could use to perturb a specific neural circuit in human patients? That's, since we technically, I don't know if we are using it, but instead of using dreads, how might we be able to do so in humans?
Well, people have tried to use a responsive neurostimulation where you put the electrodes in specific parts of the circuit. And that's a strategy that is in patients right now in a lot of contexts. I'd like to emphasize that I have a PhD. I don't have an MD. And so I'm not providing any medical advice to anyone here. So, but responsive neurostimulation is one strategy. I might emphasize that the dreads themselves are human receptors. And so it is not as foreign a substance as putting an algal protein like optogenetics into patients. And then when patients get worked up pre-surgically, that tissue will come out anyway. So I think there's the opportunity to try out some of these strategies in patients who will have that hippocampus resected anyway. And so it's not as far afield as you might think to do some translational efforts to explore whether this is going to be useful or not.
Nice. Okay, we are hitting that mark. We do have another question from Carl Broker, but I think we're going to save your question for the whole panel of Q&A.
Thank you very much.
I believe, yeah, thank you. I believe Ethan is now coming on.